Hello everyone. Oh, wait, I should have said Assalamu alaikum. Okay, from the beginning. Assalamu alaikum everyone. I'm happy to see you all here. So this video is special for the biology students. So today we're gonna talk about Alzheimer's disease, which is quite hard to pronounce, right? So let's start. So it's part 1, the hallmarks of AD. So Alzheimer's disease disrupts critical metabolic processes that keep neurons healthy. These disruptions cause nerve cells in the brain to stop working, lose connections with other nerve cells, and finally die. The destruction and death of nerve cells causes the memory failure, personality change, problems in carrying out daily activities, and other features of the disease. So, the brains of people with AD have an abundance of two abnormal structures. The first is amyloid plaques, and the second is neurofibrillary tangles. They're made of misfolded proteins. So this is especially true in the certain regions of the brain that are important in the memory. And also, there is a third main feature of AD, which is the loss of connections between cells. This leads to diminished cell function and cell death. So, now we talk about amyloid plaques. Amyloid plaques are found in the spaces between the brain's nerve cells. They were first described by Dr. Alois Alzheimer in 1906. That's just fun fact. Okay, so plaques consist of largely insoluble deposits of an apparently toxic protein peptide or fragment called beta amyloid. But we know now that some people develop some plaques in their brain tissue as they age. However, the AD brain has many more plaques in particular brain regions. We still do not know whether amyloid plaques themselves cause AD or whether they are a byproduct of the AD processes. But we do know that genetic mutations can increase production of beta amyloid and can cause rare inherited forms of AD. So, how AP develops into beta amyloid plaques? So here is 4 figures that you can see. And the first one is amyloid precursor protein. The starting point of amyloid plaques is one of many proteins associated with the cell membrane, the barrier that encloses the cell. As it's being made inside the cell, amyloid precursor protein becomes embedded in the membrane, like a toothpick stuck through the skin of an orange, as you can see in the figure 1. And a number of cell compartments, including the outermost cell membrane, specific enzymes snip or slip a amyloid precursor protein into discrete fragments. So, in the figure 2, the remaining amyloid precursor protein fragment, still tethered in the neuron's membrane, is then cleaved by gamma secretase at the end of the beta amyloid segment. The smaller of the resulting fragments also is released into the space outside the neuron, while the larger fragment remains within the neuron and interacts with factors in the nucleus. In the harmful pathway, beta secretase first sleeves the APP, which is amyloid precursor protein molecule, at one end of the beta amyloid peptide, releasing SAPP beta from the cell. as you can see in the figure 3. Gamma secretase then cuts the resulting APP fragment, still tethered in the neuron's membrane at the other end of the beta-amyloid peptide. Following the salvages at the end, the beta-amyloid peptide is released into the space outside the neuron and begins to stick to other beta-amyloid peptides, as you can see in the figure 4. These small soluble aggregates of 2, 3, 4, or even up to a dozen beta-amyloid peptides are called oligomers. Specific sites of oligomers may be responsible for reacting with receptors on neighboring cells and synapses, affecting their ability to function. It's likely that some oligomers are cleared from the brain. Those that cannot be cleared clump together with more beta-amyloid peptides. As the process continues, oligomers grow larger, becoming entities called protofibrils and fibrils. Eventually, other proteins and cellular material are added, and these increasingly insoluble entities combine to become the well-known plaques that are characteristic of AD. For many years, scientists thought that plaques might cause all of the damage to neurons that is seen in AD. However, that concept has evolved greatly in the past few years. So here you can see what we just talked about. So it is the process of beta-amyloid plaque formation. So here APP, which stands for amyloid precursor protein. This protein is located in the cell membrane, as you can see, and then this one is beta secretase and gamma secretase. These enzymes sleeve APP into smaller fragments. Here, as you can see. And beta secretase cuts APP into first, and gamma secretase further processes the remaining segment to release beta amyloid peptides. And here is the beta amyloid peptide. These fragments aggregate outside the cell, and these are oligomers, smaller clusters of beta amyloid peptides, and these are fibrils, the largest structures formed by oligomers binding together, and this is plaque, the dense deposits of fibrils, the characteristic of Alzheimer's pathology. The second hallmark of AD, also described by Dr. Alzheimer, is neurofibrillary tangles. Tangles are abnormal collections of twisted protein threads found inside nerve cells. The chief component of tangles is a protein called tau. Healthy neurons are internally supported in part by structures called microtubules, which help transport nutrients and other cellular components such as neurotransmitter-containing vessels from the cell body down the axon. Tau, which usually has a certain number of phosphate molecules attached to it, binds to microtubules and appears to stabilize them. In AD, an abnormally large number of additional phosphate molecules attach to tau. So here you can see microtubules serve as a transport system for cellular material, such as neurotransmitter vessels, maintained by normal tau protein, which stabilizes the microtubules. But in diseased neuron, hyperphosphorylated tau, abnormal chemical changes cause tau proteins to detach from the microtubules. These detached tau aggregates disrupting cellular function, as you can see here, and tau protein binds to microtubulates to maintain their integrity and enabling efficient intracellular transport and healthy neuron. But in the diseased one, Without tau stabilization, tubules break down. This leads to disrupted intracellular transport, impairing neuron function. Uh, so the cell experience damage. Accumulation of tau aggregates contributes to neurofibrillary tangles, a hallmark of Alzheimer's disease. This process leads to neuronal death and loss of communication.